I'm very excited to introduce to you my colleagues in just a moment, and I'm confident you'll find value in the information we will share with you tonight. But before we begin, please be advised that the content of these presentations is not intended to be medical advice, and that the viewers should consult their physician should they have any questions. The viewers should not rely on information contained in this presentation webinar for immediate or urgent medical needs. Additionally, if you think you may have a medical emergency, call your physician, go to the nearest emergency department, or call 911 immediately. Never disregard professional medical advice or delay seeking care because of information contained in this presentation webinar. Again, we're so pleased to have you with us tonight. Before I introduce them, I want to remind you that after we hear from our speakers, we'll have the opportunity to have your questions answered by these experts. We invite you to type your questions into the Q&A box located at the bottom of your screen. So I'm pleased to introduce Dr. Abate-Daga. Dr. Daniel Abate-Daga is a senior member in the Department of Immunology at the Moffitt Cancer Center. So, Dr. Abate-Daga, please take it away.
All right. Thank you, Dr. Pow-Sang, for the introduction, and welcome, everyone. I've been working at Moffitt for almost 10 years now, and I'm a PhD scientist doing lab-based research in the field of immunology, in particular, trying to find ways to tweak the immune system in order to allow it to detect and kill cancer cells. So today I will try to illustrate with some examples the research that we are conducting in the lab, some of which is being now translated into patient care within the scope of a clinical trial. And I will also discuss some ideas for the future moving forward on these topics. So if we move to the next slide, please. Here's a very brief introduction to the type of technologies that we work with in the lab. So as I mentioned before, most of our efforts are designed to try to educate or train the immune system so it can itself recognize and kill tumor cells. So you know that immune cells in the body are very proficient in killing cells that are infected or in neutralizing viruses or removing bacteria from the body. And they do so on the basis of recognition of elements or traits in those foreign agents that are clearly different from the body's own cells. So the immune system is trained to ignore the healthy cells and to focus on elements or organisms that it identifies as being foreign. Now, because cancer cells are a transformed cell derived from healthy tissues, it is a little bit more difficult for the immune system to naturally recognize them. It's not impossible, but it's more difficult. So what we do in the lab and in clinical trials is to take those immune cells from the blood, showing here to the left of this diagram, these rounded shapes in yellow color, which are lymphocytes. These are the main effectors, the main actors in this in this particular process, because they are able to want to kill target cells. When they recognize that there's something off with them, that they're foreign or they are sick, they're very efficient in removing those. What they lack typically is the ability to recognize tumor cells as something that they should kill. So we introduce genetic material in this, that is what is represented in this diagram, and through viral or non-viral insertion of genes into these T cells. And this genetic manipulation is intended to train them or to show them how to detect tumor cells. Now, the elements that we introduce through this genetic manipulation that will allow the cells to perform the function, they come in different flavors, so I will just mention them. You may have heard about CARs, or chimeric antigen receptors. You may also have heard recently, because there was a recent FDA approval for a new product based on a TCR or T-cell receptor. All of these molecules serve the same purpose, which is, again, allowing the T-cell to recognize what they should kill. The main difference has to do with the structure of those receptors and the type of targets that they will be able to recognize. But in any case, after we introduce the genetic material to give instructions to the T-cells on how to make those receptors that they will use to detect cancer cells. These cells that were manipulated out of the body will then be infused back to the patient in large numbers. They will circulate around the body, and they will find the target cells and eventually kill them. That's the expectation. One important aspect of this type of therapy that is called collectively adoptive cell therapy or adoptive immunotherapy is that, unlike a drug that will be given and will perform an action and then will be excreted by the body and or metabolized, the T cells, or at least a fraction of them, will have the ability to persist in the body for a long period of time. We have seen some cases in which patients have a relevant amount of gene-modified T cells in their body 7 years after they receive the treatment, meaning that they have the potential of continuing to patrol against tumor cells that may be left behind or may arise later on. And this is only when one administration of the treatment. Next slide, please. So here's a video to illustrate what happens at the microscopic level. The cyan or bluish circles that you see, these are T lymphocytes, the good guys that were genetically modified to express a CAR. The green circles are the bad guys. Those are the cancer cells. In this particular movie, the cancer cells are not prostate cancer. This is another type of cancer called Burkitt's lymphoma. The main difference is that the prostate cancers will be much bigger than those. But essentially, the series of events that are important are very similar. The T cells in blue will be floating around. Eventually, they will encounter cells that they will be able to recognize through the chimeric antigen receptor. If that recognition occurs, they will stick to them like a Velcro, and eventually will release chemicals that will cause the death of the target cells. Here in this particular video, we can visualize this dying tumor cells as based on the color. So these cells were stained so that when the cells are dying, they turn red. So you can see in the middle of the image that there's one lymphocyte attaching to actually two cells, two bad guys, and it kills one and it moves on to the second one to continue to perform its action. So this is estimated that each of these lymphocytes have the ability to take down around thousands of cells, of target cells. So it's really an efficient way of killing tumor cells, if you think about it. Next slide, please. So more specifically, talking about prostate cancer, one of the projects that we've been working on for a number of years now in the lab has to do with how to improve the efficacy of these therapies. In particular, talking about CAR T cells, there's now a number of products that have been approved by the FDA, and they're working quite well in the treatment of certain types of cancer, in particular hematological malignancies, so lymphomas, leukemias, and myelomas. But for reasons that we don't fully comprehend yet, these treatments are not as successful when they need to treat solid tumors, including prostate cancer. So I've been working on different ideas in the lab to creatively improve upon the current technology so that they will start being more effective. And one of those ideas has to do with the use of gamma delta T cells. So within the lymphocytes, the T cells that you have in the body, there are different subtypes. One of them is called gamma delta, which is it makes up for 5% of all the T cells in the circulation in the blood. So it's a rather rare population. It's not what most of the current products are based on, but we found that they have some very interesting biological properties that we could use to implement a better treatment. And in particular, one of those properties that we are interested in is the ability of these cells, unlike the most common alpha beta T cells, to interact with a drug called soledronate, which is represented in the left-hand side of this slide. And this drug is important because patients who have a certain type of prostate cancer, in particular when the disease has spread to the bone and is no longer responsive to conventional therapies, patients often receive this drug. They receive it not to kill the cancer cells, but to prevent the loss of bone, which could be caused by the presence of the tumor cells in the bone or even by the treatment that they have received before. So This is a drug that is used for a certain purpose, and we are trying here to, in a way, repurpose that drug or teach this drug how to serve a different function, which is to boost the activity of the T cells. This can only be possible, as I say, when we use gamma delta T cells. So we tried to test this concept in the lab, where we generated CAR T cells. So they are genetically modified to recognize one molecule, but we thought that because we're using this gamma delta T cells who also interact with zoledronate, we can increase the efficacy of that killing event. I probably don't need to introduce prostate cancer to this particular audience, but I wanted to just highlight that every year in the US, there's 300,000 new cases. So it affects a vast amount of people. And this event of metastasis to the bone is quite frequent, unfortunately. And at that stage, the treatment becomes more complicated. So there's clearly a need for therapies that are specifically designed to treat this particular condition of prostate cancer. In the bottom right-hand side of the slide, what I illustrated is how this would work in the clinic if it works. And I'm going to show you some experimental data soon. But what we envision is that these patients that receive the therapy will first receive an infusion with the drug, zoledronate, as they would normally for the other purposes. And then after a certain period of time, where this drug will accumulate in the bone but will be removed from the rest of the body, we will give the gamma delta CAR T cells. And this delay is important because we don't want these CAR T cells to be activated by the drug everywhere, because it could cause toxicity. So by waiting for this time, we allow it to just concentrate in the bone, which is where the drugs typically stay for a long period of time, disappear from the rest of the body, and at that point give the CAR T cells. So they will, and the two elements of this therapy will find each other inside the bone and maximize the effect there. Next slide, please. In this slide, we wanted to summarize very briefly and without boring you with a lot of experimental data, what do we observe in mouse experiments? So in vitro, which is where we typically start testing this concept, everything looked very promising. Our hypothesis is that the two elements will help each other, and with a combination therapy will achieve maximum effect, was proving to be the case. But most importantly, when we tested this in mice, we saw that there was a certain benefit of giving this combination therapy. So what I'm showing you in the left-hand side are pictures of the legs of mice, experimental mice. These mice were injected with tumor cells inside the bone, human tumor cells, and this is what is shown with those bluish and greenish clouds. So the bigger the cloud, the bigger the tumor. This is measured by a technique called bioluminescence. So on each row has photos of the same mouse at different time points. Each each column has different mouse receiving different versions of the treatment at the same time point. So for instance, the photos on the top row show the same mouse that received no treatment over time. And as you can see, the clouds, the green and blue clouds grow over time, and eventually the mice succumb to the disease. At the very end, at the bottom of this diagram, you will see the mice that receive the combination therapy. At day 0, they have a detectable tumor, but then after they receive the therapy, you see that cloud disappears. And for the most part, it stays away throughout the course of this experiment, going up to 100 days. And most of these mice actually did not have any detectable disease at the end. If we apply one element of the treatment but not the other, and that's what's represented in the rows in the middle, we achieve in some cases some level of effect, but it's not as pronounced or as durable as when we have the combo therapy. So the summary of all the mice in this experiment is shown in the graph in the middle of the slide, in case you're interested, with the dark red curve representing the growth of the tumors in the mice that received the treatment, which was much lower than with the controls. One last aspect that I would like to highlight that was a very interesting finding from this experiment is that the treatment not only reduced the size of the tumors, but it also helped with preserving the bone. You know, if you have a tumor growing inside the bones, this, as I mentioned before, this causes a deterioration of the bone and bone loss, which is problematic. But if we treat these mice with this therapy, we prevent that process, and this is illustrated by the CT or computerized tomography images in the right-hand side of the picture. The mice, the images that you see here is a reconstruction of the tip of the bone where the tumor was located. In the control mice, which did not receive any treatment, most of the bone is completely gone because the tumor is growing very aggressively. But in the gamma delta CAR plus ZO, in the bottom right-hand side of the image, you see that we have a very robust bone structure, which was, again, the result of removing the tumor and potentially having other protective effects. Next slide, please. So based on these results, we propose to start a clinical trial. We got permission from the FDA last year, and we opened the clinical trial already. This is led by Dr. Jin-Song Zhang, who is a treating physician and principal investigator. And I'm involved in the trial as a translational PI, and I'm in charge of analyzing the samples from the patients to keep learning about the biology of this cancer and the T cells themselves, so we can make them better for future therapies. At the bottom, I'm not going to spend too much time here, but in the bottom side, left-hand side of the picture, there's a diagram showing how the therapy works. Cells are collected from the blood of a patient. That's what's indicated at day -14 with a leukapheresis sign. It takes about 2 weeks to make the cell product. And at day 0, where it's labeled as CAR T-cell infusion, is where the patients receive the cells, having already been treated with zoledronate. And they are monitored for a certain period of time to make sure that the therapy doesn't cause any toxicity. And we are hoping that we will see also some clinical responses. We have treated 2 patients so far, and we plan to treat over 10 patients. So stay tuned for more results in the next few months or years. Next slides. Some ideas for the future. As we advance with the clinical testing, hoping that we will see some clinical benefit, we are already thinking about the next steps. And we are exploring some ideas. That's where I listed at the bottom right-hand side, molecular switches, strategies to enhance CAR expression, and immunobackpacks, which are technologies that are developed by some of them by my lab, some others by other collaborators. At Moffitt, for instance, the immunobackpack is a very cool idea developed by the bioengineering department, in which we can load onto these CAR T cells additional molecules that will help them perform the function. But that cannot be incorporated or taught to the T cells how to make them themselves, because they are too toxic or they don't have the capability to do so. So we can chemically bind some molecules onto the T cells. The T cells will carry them around, bring into the tumor, and release it only then. And it's a very interesting platform developed by Dr. Michael Dunn at the bioengineering department. So with a combination of this in vitro and in vivo animal modeling, plus all the information that we learn from the clinical trials, we hope that we will continue to advance these technologies into clinical practice. And we can skip the next slide and go directly into the acknowledgement. So I just wanted to highlight, without naming everyone, that this is the work of a concerted effort with multiple collaborators at Moffitt. This work has been supported by multiple sources of funding, including the Department of GU Oncology and Dr. Pow-Sang through his leveraging some of the philanthropic donations that he has secured. Also, federal agencies have funded, and we continue to rely on these sources to keep advancing these technologies, hoping to impact clinically the patients that we see at Moffitt in the near future. And I will stop there, and I will look forward to having an interesting discussion later on. Thank you.
Thank you, Dr. Abate-Daga. I'd like to introduce now Dr. Nagi Kumar. She's a senior member in the Cancer Epidemiology Program, Genitourinary and Breast Oncology Department. Pleasure to have you, Dr. Kumar. Please go ahead.
Thank you. Thank you, Dr. Pow-Sang, and that was a great presentation, Daniel. I wanted to share with you our initiative here at the Moffitt Cancer Center based on our work in cancer chemoprevention. We use a very novel approach to cancer chemoprevention. I want to share that with you today. Next slide, please. So what is cancer chemoprevention? Cancer chemoprevention is the use of botanicals, biologicals, synthetic as well as non-viral vaccines, as well as biological chemical agents to reverse, suppress, or prevent either the initial phase of carcinogenesis, or how cancers are formed, or the progression of an already existing cancer to the more advanced stages of cancer. And some of us also call that secondary chemoprevention, where we focus on individuals who have been diagnosed with cancer or pre-malignant lesions that may progress to more invasive cancers. The one Daniel was talking about before, invasion into the bone and other organs of the body. This strategy aims to limit the development and progression of malignant lesions. Next slide. So why is this an important opportunity? Because disease progression from early to later stages or metastatic disease involves a stunning increase in genomic complexity. And there's a lot of cell signaling redundancies that are very difficult to identify and treat. So a promising approach is to focus on the earliest manifestation of cancer and not wait till it becomes metastatic or more difficult. I always call these earlier stages of cancer as much dumber than the later stages of the disease. So pre-malignancies and localized cancers are simpler in their genomic aberrations, and there's an increased opportunity for us to pinpoint what is causing these early stages of cancer, and there's an opportunity for us to develop agents to target these early stages and prevent it from progressing into the more complex stages of the disease. Next slide, please. So if you look at this slide, in the past 2 decades or so, there's been so many discoveries about what causes cancer. And as you can see in this picture, it's called the hallmarks of cancer or hallmarks of carcinogenesis, or what is needed to happen for cancers to occur. And in this diagram, this is the latest published in 2022 by Hanahan and his colleagues, where you see every hallmark that has to happen, as well as some emerging or newer ways we know that cancers are formed, as well as those mechanisms that enable cancers to form. So this has guided much of our work. Next slide, please. Much of our work in discovering specific drugs that target these hallmarks of cancer. So we have a deep pipeline of drugs that have been discovered that are used as chemotherapies or immunotherapies that specifically target these hallmarks of carcinogenesis. Next slide, please. But what is the challenges that we face when we are thinking about targeted therapies for prostate cancer? We know that prostate cancer is a complex heterogeneous disease, and we all know that resistance to these targeted therapies occur, and disease recurrence is the problem. And we know that the central driving forces of prostate carcinogenesis is not just one hallmark, but a diverse set of hallmark capabilities. So it's just not a hormonal influence. It's not just the androgen receptor signaling, or it's not just a tumor suppressive activity. So it's not just any one of these hallmarks. It's several hallmarks, and we are continuing to identify. many other hallmarks as we go along. So the molecular complexity is so diverse that it demands concurrent inhibition of multiple hallmarks at the same time to prevent disease progression and disease recurrence. Next slide, please. So in our chemoprevention program, our approach is a broad spectrum approach, meaning our goal for cancer chemoprevention is to utilize a very systematic, broad spectrum approach that involves combining multiple botanicals, biologics, or repurposed agents that can produce a robust concurrent inhibition of multiple hallmark-associated pathways in targeted tissue or the microenvironment. So we are addressing, by doing this, we are addressing the underlying biology of carcinogenesis, enhancing the bioavailability and half-life of the agents that we are combining to target these cancers with minimum toxicity that can be used for a long period of time. As you can see, we are targeting earlier and earlier stages of cancer or localized stages of cancer, so we can use these drugs for a longer period of time without developing resistance. And we are targeting these drugs in high risk populations or exceptional risk populations. Next slide. So the other opportunity is that prostate cancer doesn't happen overnight, or it doesn't progress overnight. We know that the disease has a long latency period, so it takes a long time to progress from an early stage or a localized stage to a more advanced stage. So we have this opportunity to stop it from moving from a localized prostate cancer to the more advanced stage disease. So this is another opportunity. Next slide. So can you please? So we use a very systematic process by which can you please click as many times as possible? Yeah, so we get the whole. So as you can see from this diagram, this is our approach. And I think Daniel also talked about this. We are very systematic in how we develop our drugs for chemoprevention. We first tested in vitro in cells, in prostate cancer cells, in this example. And then if we get a signal there and we understand the mechanism by which it can modulate carcinogenesis, we take it to a preclinical model or a mouse model. And if it works there in a mouse model that's very relevant to, say, prostate carcinogenesis, then we take it to a human model or a human clinical trial. And if it works there, then we will take it to a phase 3 clinical trial. And if it works there, we take it to clinic. And this is the systematic way of developing any drug. Next slide. So in this process, we have identified, not just our team, but others around the country, have identified several agents and have been working with several agents. And most of them have not panned out when it moved to animal to human. But there is one agent that we got very lucky with, and that is a green tea catechin. Green tea catechins are common. It's very commonly consumed in Asian populations, especially in China, Japan, Korea, and other countries. And there are 4 major catechins present in green tea. And one of those is called epigallocatechin gallate, or EGCG. There are 3 others, but those other 3 catechins in green tea are not bioavailable. And if you drink a lot of green tea and we check your green tea concentrations in your plasma or serum in blood, you'll only find EGCG, that one catechin, and not the others. So we know now that EGCG is the most bioactive catechin in green tea. Next slide. And as we also know that countries like China and Japan and Korea have a much lower incidence of clinically evident prostate cancer compared to the Western world. And those were the initial studies that gave us these trend observations of why we picked green tea catechin to assess in laboratory studies as well as animal studies. We worked with green tea catechins, both natural forms of catechins as well as synthetic forms of catechins in our drug discovery labs. And we have found that green tea catechins are very potent inhibitors, proteasome inhibitors, which is one of the pathways that causes perturbation in the cell cycle that can lead to cancers. So next slide. I just want to, next slide, please. So in this picture, we bring back those hallmarks of carcinogenesis. And we saw not only our team, but several teams around, research teams around the country have shown that EGCG, that one specific catechin that I've been talking about, mitigates or inhibits several hallmarks associated pathways to carcinogenesis, specifically prostate carcinogenesis. In this, you can see it affects 8 pathways or 8 hallmarks of carcinogenesis. And we published this in seminars in cancer biology. And this became the basis for us to take it to the next stage. Next slide, please. So then we took it to animal studies. Not only our group, but others did the same. And consistently, there was a reduction in tumor incidence as well as tumor volume, and also a reduction in progression, aggressive progression in prostate cancer in animal models. But the thing, I mean, you'd think, okay, EGCG is the only bioactive component of green tea, and why not just use EGCG as a drug and not worry about the entire green tea component? However, the animal studies that used EGCG alone versus green tea as a whole compound didn't show good bioavailability, or they showed relatively low oral bioavailability compared to tea catechins, slow absorption, as well as high metabolic clearance by the liver. And also, EGCG administered only as part of green tea catechins had robust bioavailability and reduction in tumor volume. So there was some kind of properties of the other catechins and other substances present in the green tea compound that was useful rather than just the EGCG itself. Next slide, please. So in our study, we kind of repeated those studies that were done using a mouse model also, and we found the same results. We found that 100% of the mouse drinking water developed metastatic prostate cancer, compared to 13% consuming the green tea catechins at the highest dose. So we used only green tea as the beverage of choice for these mouse the entire time, or their entire lifetime. And higher grade tumors developed in controls versus the treatment groups. And we published this in Journal of Cancer Prevention Research. So I'm not showing a lot of data, but it was also safe to administer the volume of green tea catechins we administered to these mice. So we had signal both from the cellular studies as well as from these animal studies. And our next step was to take it to humans. Next slide, please. So we took it to men who had early stages or precursor stages of prostate cancer. Others also, there was a group in Arizona, University of Arizona, that also tested the bioavailability as well as tolerability of the doses of green tea catechins in 200 to 1200 milligrams a day, and it was found to be safe. In our studies, we observed a lower incidence of progression from these precursor lesions to prostate cancer. We also found a significant reduction of serum PSA in our patient population consuming the green tea catechin. Again, here we developed a standardized drug of green tea catechins that was approved by the FDA for use in our clinical trial. And there were no adverse events that were different between the group that got the catechin capsules compared to the group that got the placebo. So these were randomized clinical trials. Next slide, please. So we published quite a bit in this area. We published our work in animals and the cells, and as well as in the early precursor stages of prostate cancer called high-grade PEN. I'm sure some of you would have heard about it. Next slide, please. And based on our work, as well as that of others, we summarize that green tea catechins, specifically this EGCG, has been shown to inhibit several cancer-related proteins, contributing to modulation of several hallmarks of cancer compared to benign controls. And this is specifically in prostate cancer. We have shown that EGCG is observed to be a potent inhibitor of prostate cancer carcinogenesis through multiple mechanisms, as it hits multiple hallmarks of carcinogenesis. It accumulates in plasma as well as in prostate tissue. That's important. Does it get to the organ? And we and others have showed that it does. It reduces serum PSA significantly, and it reduces rate of progression to prostate cancer. And these are available in standardized formulations that we can use as research scientists in our early phase clinical trials. And there are no toxicities at the doses, as I showed you, 200 to even 1200. So we had all this evidence, and now we move to the next stage of clinical drug development, that is focusing on men with localized prostate cancer with Gleason 3+3 and 3+4. 3+4, not all 3+4, those who had a lower risk of progression, other target population we selected. Next slide. So we have two trials that are currently ongoing and funded by the National Cancer Institute. It is a phase 2 clinical trial of these same catechins. We are using a higher dose of the capsule. It's 800 milligrams instead of 200 that we used in the high-risk population, because this is men with early stage cancer or localized disease who are placed on active surveillance. Currently, there is no treatment for men who have low-grade prostate cancer or Gleason 3+3. They are placed on active surveillance. And this is the target population we are conducting this randomized clinical trial on. We have about 90 subjects who are on the trial. Next slide. We also wanted to make it available nationwide. So we were funded by the National Cancer Institute to run the same trial nationally. In this, we are also looking at, even in the previous trial, we are looking at validating the mechanisms that we observed in our in vitro work or in our laboratory studies. In this study, we have opened the study nationally, and we are also recruiting men from urology practices and not just from comprehensive cancer centers. Next slide. So ultimately, our goal is to conduct these two clinical trials to comprehensively and rigorously evaluate the safety, effectiveness, as well as the mechanism by which these catechins impact clinical and biological intermediate endpoint biomarkers implicated in prostate cancer. This will inform ultimately our phase 3 clinical trial. Again, this is targeting men who are on active surveillance for diagnosis of prostate cancer. Next slide, please. We have the same approach we are using with other agents. The same agent looks very promising in chronic lymphocytic leukemia as well as in multiple myeloma. And we have started the preliminary work on these two targets. Next slide. I'll be done in 30 seconds. We have other drugs using the same approach of targeting multiple hallmarks of carcinogenesis. We have one in bladder cancer also, which is a sulforaphane, another a breast study as well as a lung study. Next slide. I just wanted to thank our patients and their families. They are very optimistic and enthusiastic participating in our clinical trials, which are intervention trials that are harder to go through 2 years of in a clinical trial, and for them to continue to inspire our work. And many thanks to our team at Moffitt as well as our collaborators. Thank you.
Thank you, Dr. Kumar. That was a great presentation. Now I'd like to introduce one of our partners here at Moffitt, Mr. Michael Quinn. Mike is with the Moffitt Foundation, and he partners with the Genitourinary Program to ensure that we receive the research dollars that we need to get this amazing clinical research off the ground and make advances in cancer treatments. If you'd like to learn more about how you can help, please contact Mike to find out more. Mike, would you like to say some words?
Sure. Thank you, Dr. Pow-Sang. In just a few minutes, we will return to have the Q&A portion of the webinar. I know that's the exciting part of tonight, so that our panelists will be able to answer those questions you have started to ask. And if you do have some more questions, we encourage you to please put those in the Q&A component of this webinar. I would like to take some time to thank you for joining us this evening. We understand that your time is valuable, and there are numerous other commitments you could be attending to on this Thursday evening. And your choice to join us is deeply appreciated, and we truly are grateful for your support. I also want to thank Dr. Pow-Sang, Dr. Abate-Daga, and Dr. Kumar for providing such good insight into the work being done in prostate cancer research. Additionally, I want to thank my colleagues Brielle Humphreys and Stacy Price for managing this webinar behind the scenes tonight. So thank you for that. Tonight's distinguished experts that we've heard from not only make a significant difference for local patients, but they also contribute to advancements with a global impact. There are several ongoing research projects in genitourinary oncology, including specific studies on prostate cancer that are in need of funding. If you are interested in exploring opportunities to provide philanthropic support for these vital research projects, please feel free to contact me at your convenience. Thank you again for your participation in tonight's webinar, and I will now turn the floor back over to Dr. Pow-Sang to begin the Q&A section. Dr. Pow-Sang?
Thank you, Mike. We appreciate all that the Foundation does with philanthropy to make our research possible. Now we are going to open the session for questions. We already have a lot of questions. That's impressive. I will start reading the questions and invite the faculty members to respond. So I'm going to go ahead and start looking at the questions. And the first question, I'm going to open it up. The first question is, what % of new diagnosis of prostate cancer metastasize to the bone? I can answer that. If we take all the newly diagnosed men with prostate cancer in a year. About 80% present with early prostate cancer. And that's important because the earlier you diagnose cancer that needs to be treated, the better the outlook. Cancers that are locally advanced are probably about 10%. And then cancers that present with metastasis to the bone are probably about 10%. And these are men that could benefit with the treatment that Dr. Daniel Abate was mentioning. After going with initial treatment, who had failed hormonal therapy, who had failed chemotherapy. So these are very selected patients with cancer that has not responded to previous treatments. There's another question here for Dr. Abate regarding what are the side effects with the treatment with CAR T cells, Dr. Abate?
Yes, it's too early to tell at this point. So we expect to learn from the ongoing clinical trial. So stay tuned for more updates on that. What I can comment on is based on other clinical trials for similar products. So the typical side effects or adverse events associated have to do with two processes mostly. One is called cytokine release syndrome, which is produced by the activation of this large number of cells going into the body that can cause some alterations in the fluid management of the body. And over the years, the physicians have learned how to treat this condition. It doesn't happen to all of the patients, but now there are very clear protocols on how to manage this type of toxicity. One interesting aspect of this, the CAR T based on gamma delta T cells that we are testing now, is that we anticipate or we hypothesize that this type of complication will have a very low incidence because of some of the interesting biological properties of the cells. They are very good at killing the target cells that we put in front of them, but they do so without causing a lot of trouble around it. So this process of cytokine release syndrome is caused by the production of cytokines. It's a type of chemical compound that the T cells can produce, and not only T cells and others as well. But we hope and we expect, based on our preclinical data, that this will be less of a problem in our setting, because gamma deltas, unlike the most common type of alpha beta T cells, they're cleaner in a way in their ability to kill the target cells without releasing a lot of chemical compounds around. So again, we will learn more about it as we advance with the trial. But thank you for the question.
Yeah. Thank you, Dr. Abate. There's another question here. Does one plan to try treatment for someone with PIRATS4 cancer? When a little 101 on PIRATS4, that's a grading to look at lesions on prostate MRI. These are very early cancers. This is not the space for this type of treatment. Just to give you an idea, when a patient presents with an elevated PSA, which is probably 90% of the presentations at present time, the next test is to either molecular testings with blood or urine to check about the probability of having significant prostate cancer. And if there's a higher probability, the next test is a prostate MRI, magnetic resonance imaging, and the test might be negative, or it might show some spots in the prostate. And depending on how significant the spots are, one gives a PIRATS grading. That's a grade which goes from 1 to 5. And the 1 and 2s are considered negatives. The 3 are on the fence, and the 4 and 5 have a higher probability if there's a cancer. So a PIRATS4 or a 5 lesion in that grading system doesn't necessarily mean that there's a cancer. One does a biopsy, and it could be negative because many benign things can give an appearance of a PIRATS4, and sometimes it's positive. And if the biopsy is positive, then we have to determine if the cancer is what is called a significant cancer that needs to be treated, because it's important to know that many of the early cancers that are diagnosed nowadays do not need to be treated when we place them on active surveillance. And then we do additional testings. If the cancer is too aggressive, then we consider a treatment. There was another question here regarding for stage 4, which is metastatic cancer that has spread to the bones, that has progressed beyond what is called Xtandi. Xtandi is a hormone type of treatment, are called androgen path inhibitors. They are used together with injections or oral medications to block male hormones. And what happens when the cancer progresses, eventually the treatment is effective for about 2 years or 5 years, but then the cancer breaks through, and then there are other treatment options. There is chemotherapy, and there are 2 chemotherapy agents that are used if that is the case. And if the cancer progresses, there's immunotherapy, and then there's cell therapy, like Dr. Abate was discussing. So one of the significant things that have happened in the last 10 years is that only 10 years or before that, there was only one treatment, we got hormonal therapy. Now there are many hormonal therapies, and if the cancer progresses, there are a sequence of other treatments like chemotherapy, immunotherapy, and the treatment like what Dr. Abate was mentioning. There's a question here regarding irreversible electroporation of the prostate, which is a newer focal treatment for prostate cancer. When one diagnoses the cancer, as I was mentioning before, most of the cancers are early localized. Some don't need to be treated, but some do. And the conventional treatment is to remove the prostate or to take radiation therapy. But there's a proportion of cancers, it's believed that it's about 10 to 20% that are very localized, and we have technology to actually localize the cancer within the prostate, see exactly where it is, and treat it with what is called focal therapy, for which irreversible electroporation, or IRE, is one of the ways. One put needles into the prostate with anesthesia through the skin between the testicles and the rectum, and places electrodes around the tumor, produce an electrical field, and destroy the cancer that way. So there's irreversible electroporation. There's another technology called cryosurgery. We put needles in the prostate and freeze the prostate. There's another technology called high-intensity focused ultrasound. We put a probe in the rectum, it produces high-frequency ultrasound, and that destroys the cancer by heat. So many centers are using one or the other technology. Here at Moffitt, because we have a very strong program in focal therapy, we use all 3 technologies in patients in whom this is indicated. So I hope that answers that question. How do we determine the stage of prostate cancer? Is it through PSA, genomic testing? Briefly, in general, when we diagnose a cancer, we do tests, which are PSA. The PSA is very predictive of how extensive the cancer is. And one adds other tests, what are called imaging testing. One of the revolutions has been for cancers that have a high probability of being advanced. We don't do what still some people in the community do, which is bone scans to assess the bones with nuclear medicine or CAT scan. But there's a test that's been available here in the US for about 3 or 5 years, which is called PET scan, but it's a very specific PET scan for prostate cancer. It's called a PSMA PET scan, prostatic specific membrane antigen, and it's very sensitive to detect cancer spread. So depending on the circumstances, one adds tests, depending on the probability of having cancer spread, to assure that one knows how extensive the cancer is, because that drives the type of and extent of treatment that one might need. There's a question for Dr. Kumar. Who asks, someone were to regularly drink green tea, could they receive any benefit from the catechins contained within it, or does it have to be larger doses? So it's a question of dosing, Dr. Yes. Kumar.
So we've seen that in populations that consume green tea consistently, this is brewed green tea that, for example, Japanese consume all day. That's a dose that prevents them from, we know from the data that prevents them from getting clinically evident advanced cancers. So Based on that, it's harmless to drink green tea as a beverage. But I think to prevent carcinogenesis from a localized to an advanced stage, I think you need a higher dose, and that's why we developed a therapeutic drug that we are administering in these randomized clinical trials. And we also have increased the concentration of EGCG to be higher than what's naturally available in a green tea catechin. And just in relation to that, I would just want to answer another question I saw about why are the other catechins not working? Why are the other catechins not important? We don't detect them in the plasma or the serum or in prostate tissue. But as the rider indicates, there's some synergistic value of giving it in combination. So there is some value to it as it occurs naturally, and that's why we have it in a compound.
Thank you, Dr. Kumar. There were some questions regarding staging of prostate cancer. So specifically regarding non-metastatic castrate-sensitive prostate cancer, castrate-resistant, non-metastatic, and then metastatic. What happens with prostate cancer is that it is very sensitive and thrives and grows and feeds on male hormones. So the initial treatment for cancer that has spread to the system, which is called metastatic into the lymph nodes or the bones, is to first achieve what is called hormone blockade. One male blocks male hormones, and the cancer responds at the beginning, but in time, as I mentioned before, it becomes resistant. It's like a bacteria that is resistant to antibiotics, and one has to try another antibiotic, which the bacteria hasn't seen, and kills the bacteria. Same thing. When the cancer stops responding to the blockade of hormones, then one uses additional treatments, one of which is chemotherapy, as I mentioned. There are 2 chemotherapies that are approved. And if that one doesn't respond to chemotherapy, then one uses immunotherapy. And there is immunotherapy that is approved for use. There are several others that have been studied. And there's cell therapy that Dr. Daniel Abate mentioned. So I hope that answers the question. I'm going to take another question because there are so many questions, and I want to stress again that you have the link, your email, and we make every effort to respond to every single question in the next several days. So there was a question here. How the cancer metastasizes or spread. Well, usually the cancer progresses in 2 ways. One is locally. It walks through the capsule of the prostate, the envelope, and to the adjacent organs. But in the majority of cases, the cancer metastasizes, spreads out through the bloodstream of the lymph nodes. And in those cases, one uses the systemic treatments that I mentioned before. So we are close to the end. There are so many questions, and I wish we could spend more time talking about it. But again, we are very available. We're going to go through the questions and the ones we generated and get back to you all to try to answer as many questions as possible. So thank you very much for taking time to learn about prostate cancer. We are always available. We appreciate your continued support to work as we revolutionize cancer care and provide this possible convenience. Thank you very much.